Principle of inheritance and variation. Have you ever wondered why an elephant always gives birth only to a baby elephant and not some other animal, or why a mango seed forms only a mango plant and not any other plant? Given that they do, are the offspring identical to their parents, or do they show difference in some of their characteristics? Have you ever wondered why siblings sometimes look so similar to each other or sometimes even so different? These and several related questions are dealt with scientifically in a branch of biology known as genetics. This subject deals with the inheritance as well as variation of character from parent to offspring. Inheritance is the process by which characters are passed on from parent to progeny. It is the basis of heredity. Variation is the degree by which progeny differ from their parents. Humans knew from as early as 8000 to 1000 BC that one of the causes of variation was hidden in sexual reproduction. They exploited the variation that were naturally present in the wild population of plants and animals to selectively breed and select for organisms that possessed desirable characters. For example, through artificial selection and domestication from ancestral wild cow, we have well-known Indian breeds. Example, Sahiwal cow in Punjab. We must, however, recognize that though our ancestors knew about the inheritance of character and variation, they have very little idea about the scientific basis of this phenomena. Mendel's law of inheritance. It was during the mid-19th century that headway was made in the understanding of inheritance. Gregor Mendel conducted hybridization experiments on garden peas for 7 years, 1856 to 1863, and proposed the law of inheritance in living organisms. During Mendel's investigation into inheritance pattern, it was for the first time that statistical analysis and mathematical logic were applied to problems in biology. His experiment had a large sampling size, which gave greater credibility to the data that he collected. Also, the confirmation of his inference from experiment on successive generation of his test plant proved that his result pointed to general rules of inheritance rather than being unsubstantiated ideas. Mendel investigated characters in the garden pea plant that were manifested as two opposite traits. Example, tall or dwarf plants, yellow or green seeds. This allowed him to set up a basic framework of rules governing inheritance, which was expanded on by later scientists to account for all the diverse natural observation and the complexity inherent in them. Mendel conducted such artificial cross-pollination experiment using several true breeding pea lines. A true breeding line is one that, having undergone continuous self-pollination, shows the stable trait inheritance and expression of for several generations. Mendel selected 14 true breeding pea plant varieties as pairs which were similar except for one character with contrasting trait. Some of the contrasting traits selected were smooth or wrinkled seed, yellow or green seed, inflated full or constricted green or yellow spot, and tall or dwarf plants. Contrasting traits studied by Mendel in pea: stem height, tall dwarf, flower color violet white, flower position axial or terminal, pod shape inflated or constricted, pod color green or yellow, seed shape round or wrinkled, seed color yellow green. Inheritance of one gene. Let us take the example of one such hybridization experiment carried out by Mendel, where he caused tall and dwarf pea plant to study the inheritance of one gene. He collected the seed produced as a result of this cross and grew them to generate plant of the first hybrid generation. This generation is also called the Philia 1 progeny or the F1. Mendel observed that all the F1 progeny plant was tall like one of its parents. None were dwarf. He made similar observation for the other pairs of traits. He found that F1 always resembled either one of the parents and that the trait of the other parents was not seen in them. Mendel then self-pollinated the tall F1 plants and to his surprise found that in the filial 2 generation some of the offspring were dwarf. The character that was not seen in F1 generation was now expressed. The proportion of plants that were dwarf were 1/4 of the F2 plants, while 3/4 of the F2 plants were tall. The tall and dwarf traits were identical to their parental type and did not show any blending. That is, all the offspring were either tall or dwarf. None were of in between height. Similar result was obtained with the other trait that he studied. Only one of the parental state was expressed in the F1 generation, while at the F2 stage both the traits were expressed in the proportion 3:1. The contrasting traits did not show any blending at either F1 or F2 stage. Based on the observation, Mendel proposed that something was being stably passed down unchanged from parent to offspring through the gametes over successive generations. He called these things as factors. Now we call them as genes. Genes, therefore, are the unit of inheritance. They contain the information that is required to express a particular trait in an organism. Genes which code for a pair of contrasting traits are known as alleles. That is, they are slightly different form of the same gene. If we use alphabetical symbol for each gene, then the capital letter is used for the trait expressed at the F1 stage and small alphabet for the other trait. For example, in case of the character of height, capital T is used for the tall trait and small t is used for dwarf, and capital T and small t are alleles of each other. Hence, in plants, the pair of alleles for height would be capital T, small t, small t, or small t. Mendel also proposed that in a true breeding, tall or dwarf pea variety, the allelic pair of genes for height are identical or homozygous, capital T and small t respectively. Capital T and small t are called genotype of the plant, while the descriptive term tall and dwarf are the phenotype. What then would be the phenotype of the plant that had a genotype capital T small t? As Mendel found the phenotype of the F1 heterozygote capital T small t to be exactly like the capital T parent in the appearance. He proposed that in a pair of dissimilar factor, one dominates the other as in the F1 and hence is called the dominant factor while the other factor is recessive. In this case, capital T for tallness is dominant over small t for dwarfness, that is recessive. He observed identical behavior for all the other character or trait pairs that he studied. It is convenient and logical to use the capital and lower case of an alphabetical symbol to remember this concept of dominance and decisiveness. Do not use capital T for tall and small d for dwarf because you will find it difficult to remember where capital T and small d are alleles of same genes or character or not. Alleles can be similar as in the case of homozygotes capital T and small t or can be dissimilar as in the case of the heterozygote capital T small t. Since the capital T small t plant is heterozygous for gene controlling one character height, it is a monohybrid and the cross between capital T and small t is a monohybrid cross. From the observation that the recessive parental trait is expressed without any blending in the F2 generation, we can infer that when the tall and dwarf plants produce gametes, by the process of meiosis, the alleles of the parental pair separate or segregated from each other and only one allele is transmitted to gamete. This segregation or allele is a random process and so there is a 50% chance of gamete containing either allele as has been verified by the result of the crossing. In this way, the gamete of the tall capital T plants have the alleles capital T and the gamete of the dwarf small t plant have the allele small t. During fertilization, the two alleles capital T form from one parent say through the pollen and small t from the other parent then through the egg are united to produce zygote that have one capital T allele and one small t allele. In other words, the hybrid has capital T small t. Since the hybrid contain alleles which express contrasting traits, the plant are heterozygous. The production of gametes by the parent, the formation of the zygotes, the F1 and F2 plants can be understood from a diagram called Punnett Square. It was developed by British scientist Reginald C. Punnett. It is a graphical representation to calculate the probability of all possible genotype of offspring in a genetic cross. The possible gametes are written on two sides, usually the top row and left column. All possible combinations are represented in box below in the square which generate a square output form. The Punnett Square shows the parental tall capital T male and dwarf small t female plants. The gamete produced by them and F1 capital T small t progeny. The F1 plant of the genotype capital T small t are self-pollinated. The symbols plus and arrow are used to denote the female egg and male pollen of the F1 generation respectively. The F1 plant of the genotype capital T small t when self-pollinated produce gamete of the genotype capital T and small t in equal proportion. When fertilization takes place, the pollen grains of genotype capital T have a 50% chance to pollinate rate of the genotype capital T as well as of genotype small t. Also pollen grains of genotype capital small t have a 50% chance of pollinating egg of genotype capital T as well as of genotype small t. As a result of random fertilization, the resultant zygote can be of the genotype capital T small t or T small t. From the Punnett Square, it is easily seen that one-fourth of the random fertilization lead to capital T, half lead to capital T small t, and one-fourth to small t. Though the F1 have a genotype of capital T small t, but the phenotype character seen is tall. At F2, three-fourths of the plants are tall, where some of them are capital T, while other are capital T small t. Externally, it is not possible to distinguish between the plant with genotype capital T and capital T small t. Hence, within the genotypic pair capital T small t, only one character capital T tall is expressed. Hence, the character capital T or tall is said to be dominant over the other allele small t or dwarf character. It is thus due to this dominance of one character over the other that all the F1 are tall, though the genotype is capital T small t, and in the F2, three-fourths of the plant are tall, though genotypically half are capital T small t and only one-fourth are capital T. This leads to a phenotypic ratio of three-fourths tall, one-fourth capital T and plus half capital T small t and one-fourth small t. That is a 3:1 ratio, but a genotypic ratio of 1:2:1. The 1/4:1 half is to 1/4 ratio of capital T ratio capital T small t ratio capital small t mathematically considerable to the form of binomial expression ax plus by whole square that has the gamete bearing genes capital T or small t in equal frequency of half. The expression is expanded as given below. Half capital T plus half small t square equal 1/4 capital T plus half capital T small t plus 1/4 small t. Mendel self-pollinated the F2 plant and found that dwarf F2 plant continue to generate dwarf plants in F3 and F4 generation. He concluded that the genotype of the dwarf was homozygous small t. What do you think he would have got had he self-pollinated a tall F2 plant? From preceding paragraph, it is clear that though the genotypic ratio can be calculated using mathematical probability by simply looking at the phenotype of a dominant trait, it is not possible to know the genotypic composition. That is, for example, whether a tall plant from F1 or F2 has capital T or capital T small t composition cannot be predicted. Therefore, to determine the genotype of a tall plant at F2, Mendel caused the tall plant from F2 with a dwarf plant. This he called a test cross. In a typical test cross, an organism pea plant here showing a dominant phenotype and whose genotype is to be determined is crossed with the recessive parent instead of self-crossing. The progenies of such a cross can easily be analyzed to predict the genotype of the test organism. Figure shows the result of typical test cross where violet color flower W is dominant over the white color flower W. Capital W or violet color flower capital W, white colored flower small w. Using Punnett Square, try to find out the nature of offspring of a test cross. What ratio did you get? Using the genotype of this cross, can you give a general definition for a test cross? Based on his observation on monohybrid crosses, Mendel proposed two general rules to consolidate his understanding of inheritance in monohybrid crosses. Today, these rules are called the principle or laws of inheritance. The first law or law of dominance and the second law or law of segregation. Law of dominance. Character are controlled by discrete units called factors. Two factors occur in pairs. Three in a dissimilar pair of factor. One member of the pair dominant and the other recessive. The law of dominance is used to explain the expression of only one of the parental character in a monohybrid cross in the F1 and expression of the both in F2. It also explains the proportion of 3:1 obtained at the F2. Two law of segregation. The law is based on the fact that the alleles do not show any blending and that both the character is recovered as such in the F2 generation, through one of these is not seen in at the F1 stage. Though the parent contains two alleles during gametes formation, the factor or alleles of a pair is segregated from each other such that a gamete receives only one of the two factors. Of course, a homozygous parent produces all gametes that are similar, while a heterozygous one produces two kinds of gametes, each having one allele with equal proportion. Incomplete dominance. When experiments on the P were repeated using other traits in other plants, it was found that sometimes the F1 had a phenotype that did not resemble either of the two parents and was in between the two. The inheritance of the flower color in the dog flower, snapdragon or Antihism species is a good example to understand incomplete dominance. In a cross between two breeding red flower RR, capital R, capital R and two breeding white flower plants, small r, the F1 capital R, small r was pink when the F1 was self-pollinated. The F2 result in the following ratio: 1 capital R red, 2 capital R pink, 1 small r white. Here the genotype ratio were exactly as we would expect in any Mendelian monohybrid cross, but the phenotype ratio had changed from the 3:1 dominant recessive ratio. What happened was that capital R was not completely dominant over the small r, and this made it possible to distinguish capital R, small r as pink from capital R red and small r white. Explanation of the concept of dominance. What exactly is dominance? Why are some alleles dominant and some recessive? To tackle these questions, we must understand what a gene does. Every gene, as you know by now, contains the information to express a particular trait. In a diploid organism, there are two copies of each gene, that is, as a pair of alleles. Now, these two alleles need not always be identical as in a heterozygote. One of them may be different due to some changes that it has undergone, about which you will read further on and in this next chapter, which modifies the information that particular allele contains. Let us take an example of a gene that contains the information for producing an enzyme. Now there are two copies of these genes, the two allelic forms. Let us assume, as is more common, that the normal allele produce the normal enzyme that is needed for the transformation of a substrate. Yes, theoretically, the modified allele could be responsible for production of one normal or less efficient enzyme, or a non-functional enzyme, or no enzyme at all. In the first case, the modified allele is equivalent to the unmodified allele. That is, it will produce same phenotype trait, that is, result in transformation of substrate. Yes, such equivalent allele pair are very common, but if the allele produce a non-functional enzyme or no enzyme, the phenotype may be affected. The phenotype or the trait will only be dependent on the functioning of the unmodified allele. The unmodified functioning allele which represent the original phenotype is the dominant allele, and the modified allele is generally the recessive allele. Hence, the example of the above recessive trait is seen due to non-functional enzyme or because of no enzyme is produced. Codominance. Till now we were discussing cross where the F1 resemble either of the two parents' dominance or was in between incomplete dominance, but in the case of codominance, the F1 generation resemble both parents. A good example is different types of red blood cell that determine ABO blood group P in human beings. ABO blood groups are controlled by the gene I. The plasma membrane of a red blood cell has sugar polymer that protrude from its surface, and the kind of sugar is controlled by the gene. The gene I has three allele, IA, IB and small I. The capital IA and capital IB produce a slightly different form of sugar, where the allele small I does not produce any sugar. Because humans are diploid organisms, each person possesses any two of the three capital I gene allele. Capital IA and capital IB are completely dominant over small I. In other words, when capital A and small I are present, only capital I expressed because small I does not produce any sugar. And when capital IB and small I are present, capital IB expressed. But when capital I A and capital IB present together, they both express their own types of sugar. This is absence of codominance. Hence, red blood cells have both A and B type of sugar. Since there are three different allele, there are 6 different combinations of these 3 allele that are possible, and therefore a total of 6 different genotypes of the human ABO blood groups. However, many phenotypes are possible. Do you realize that the example of ABO blood grouping also provides a good example of multiple alleles? Here you can see that there are more than 2, that is 3 alleles governing the same character. Since in an individual only 2 alleles can be present, multiple alleles can be found only when population studies are made. Occasionally, a single gene product may produce more than one effect. For example, starch synthesis in pea seed is controlled by one gene. It has 2 alleles, B, capital B and small b. Starch is synthesized effectively by capital B, homozygotes, and therefore large starch grain are produced. In contrast, small homozygotes have lesser efficiency in starch synthesis and produce smaller starch grain. After maturation of the seed, B seeds are round and the small b seeds are wrinkled. Heterozygotes produce round seeds, and so B seem to be dominant allele, but the starch grain produced are of intermediate size in B seed. So if starch grain size is considered as the phenotype, then from this angle the allele show incomplete dominance. Therefore, dominance is not an autonomous feature of a gene or the product that it has information for. It depends as much on the gene product and production of a particular phenotype from this product, and as it does on the particular phenotype that we choose to examine. In case more than one phenotype is influenced by the same gene. Inheritance of two genes. Mendel also worked with the crossed pea plant that differed in two character, as is seen in the cross between a pea plant that had seed with yellow color and round shape and one that had seed of green color and wrinkled shape. Mendel found that seed resulting from the crossing of the parents had yellow colored and round shaped seed. Here can you tell which of the character in shape in the pairs yellow or green color and round and wrinkled shape was dominant? Thus, yellow color was dominant over the green and round shape dominant over wrinkled. These results were identical to those that he got when he made separate monohybrid crosses between yellow and green seeded plant and between round and wrinkled seed plant. Let us use the genotypic symbol Y for dominant yellow seed color and small y for recessive green seed color. Capital R for round shaped seed and small r for wrinkled seed shape. The genotype of the parent can be written as capital R, capital Y, and small r, small y. The cross between the two plants can be written as in the figure showing genotype of the parent plants. The gamete capital R, capital Y, and small y unite on fertilization to produce the F1 hybrids capital R, capital Y, small y. When Mendel self-hybridized the capital F1 plants, he found that 3/4 of F2 plant had yellow seed and 1/4 had green. The yellow and green color segregated in ratio 3:1 ratio. Round and wrinkled seed shape also segregated in 3:1 ratio, just like a monohybrid cross. Law of independent assortment. In the hybridized cross, the phenotype round yellow, wrinkled yellow, round green, and wrinkled green appear in the ratio 9:3:3:1. Such a ratio was observed for several pairs of characters that Mendel studied. The ratio of 9:3:3:1 can be derived as a combination series of 3 yellow, 1 green, 3 round, 1 wrinkle. This deviation can be written as follows: 3 round is to 1 wrinkle, 3 yellow into 1 green equal 9 round yellow, 3 wrinkle yellow, 3 green round green, 1 wrinkle green. Based upon such observation on hybrid process between plant different into traits, Mendel proposed a second set of generalization that we call Mendel's law of independent assortment. The law stated that when two pair of traits are combined in a hybrid, segregation of one pair of character is independent of the other pair of character. The Punnett Square can be effectively used to understand the independent segregation of two pairs of genes during meiosis and the production of egg and pollen in the F1 capital R capital Y small y plant. Consider the segregation of one pair of genes capital R and small r. 50% of the gametes have the gene capital R and the other 50% have small r. Now beside each gamete having either capital R or small r, it should also have the allele capital Y and small y. The important thing to remember here is that segregation of 50% capital R and 50% small r is independent from the segregation of 50% capital Y and 50% small y. Therefore, 50% of small r being bearing gametes has capital Y and other 50% has small y. Similarly, 50% of capital R bearing gamete has capital Y and other 50% have small y. Thus, there are four genotypes of gametes, four types of pollen and four types of eggs. The four types of are capital R, capital Y, capital R, small y, small r, capital Y and small r, small y. Each with a frequency of 25% or 1/4 of the total gamete body. When you write down the four types of eggs and pollen on the two sides of the Punnett Square, it is very easy to derive composition of the zygotes that gives rise to the F2 plant. Although there are 16 square, how many different types of genotype and phenotype are formed? Note them down in the format given. Can you, using the Punnett Square data, work out the genotypic ratio at the F2 stage and fill in the format given? Is the genotypic ratio also 9:3:3:1? Chromosomal theory of inheritance. Mendel published his work on inheritance of character in 1865, but several reasons it remained unrecognized till 1900. Firstly, communication was not easy as it is now. In those days, this work could not be widely publicized. Secondly, his concept of genes or factor in Mendel's work as stable and discrete units that control the expression of the trait and of pair of alleles which did not blend with each other was not accepted by his contemporaries as an explanation for the apparently continuous variation seen in nature. Thirdly, Mendel's approach of using mathematics to explain biological phenomena was totally new and unacceptable to many of the biologists of his time. Finally, though Mendel's work suggested that factor genes were discrete units, he could not provide any physical proof for his existence of factor or say what they were made of. In 1900, three scientists, Devries, Correns and von Scharmark, independently discovered Mendel's result on the inheritance of the character. Also by this time, due to advancement in microscopy that were taking place, scientists were able to carefully observe cell division This led to discovery of structure in the nucleus that appear in the double and divide just before each cell division. These were called chromosome colored bodies as they were visualized by staining. By 1902, the chromosome movement during meiosis had been worked out. Walter Sutton and Theodore Boveri noted that the behavior of chromosomes was parallel to the behavior of genes and used chromosome movement to explain Mendel's law. Recall that you have studied the behavior of chromosome during mitosis, equation division, and during meiosis, reduction division. Chromosome during mitosis, equal division, and during meiosis, reduction division. The important thing to remember are that chromosomes as well as genes occur in pairs. The two alleles of a gene pair are located on homologous side on homologous chromosome. Comparison between the behavior of chromosome and genes. A occur in pairs, B occurs in pairs. Yes, segregation at the time of gamete formation such that only one of each pair is transmitted to a gamete. B segregate at gamete formation and only one pair of each pair is transmitted to a gamete. A independent pair segregate independently of each other. B one pair segregate independently of another pair. A and B. Can you tell which of these columns A or B represent the chromosome and which represent the gene? How did you decide? During anaphase of meiosis 1, the two chromosome pair can align at the metaphase plate independently of each other. To understand this, compare the chromosome of 4 different colors in the left and right column. In the left column, possibility 1, orange and green is segregating together, but in the right hand column, possibility 2, the orange chromosome is segregating with the red chromosome. Possibility 1, one long orange and short green chromosome and long yellow and short red chromosome at the same pole. Possibility 2, one long orange and short red chromosome and long yellow and short green chromosome at the same pole. Sutton and Boveri argued that the pairing and separation of a pair of chromosomes would lead to the segregation of a pair of factors they carried. Sutton united the knowledge of chromosomal segregation with Mendelian principle and called it the chromosomal theory of inheritance. Following the synthesis of idea, experimental verification of the chromosomal theory of inheritance by Thomas Hunt Morgan and his colleagues led to discovering the basis for the variation that sexual reproduction produced. Morgan worked with the tiny fruit flies, Drosophila melanogaster, which were found by suitable for such studies. They could be grown on simply synthetic medium in the laboratory. They complete their life cycle in about 2 weeks, and a single mating could produce a large number of progeny of flies. Also, there was a clear differentiation of the sexes. The male and female flies are easily distinguishable. Also, it has many types of hereditary variation that can be seen with low power microscope. Linkage and recombination. Morgan carried out several dihybrid courses in Drosophila to study genes that were sex-linked. The courses were similar to dihybrid courses carried out by Mendel in P. Some example: Morgan the hybridized yellow bodies, white-eyed female to brown bodies, red-eyed female male, and intercrossed their F1 progeny. He observed that the two genes did not segregate independently to each other, and the F2 ratio deviated very significantly from 9:3:1 ratio expected when the two genes are independent. Morgan and his group knew that the genes were located on the X chromosome and saw quickly that when the two genes in a dihybrid cross were situated on the same chromosome, the proportion of parental genes combination were much higher than the non-parental type. Morgan attributed this due to the physical association or linkage of the two genes and coined in the term linkage to describe physical association of genes on a chromosome, and the term recombination to describe the generation of non-parental gene combination. Morgan and his group also found that even when genes were grouped on the same chromosome, some genes were very tightly linked, showed very low recombination, while others were loosely linked, showed higher recombination. Group cross B. For example, he found that the genes white and yellow were very tightly linked and showed only 1.3% of recombination, while white and miniature wing showed 37.2% recombination. He studied Alfred Stratford used the frequency of recombination between genes there on the same chromosome as a measure of disadvantage between genes and mapped their position on the chromosome. Today, genetic maps are extensively used as a starting point in the sequencing of whole genome, as was done in the case of human genome sequencing project described later. Polygenic inheritance. Mendel studied mainly described those traits that have distinct alternate forms such as flower color, which are either purple or white. But if you look around, you will find that there are many traits which are not so distinct in their occurrence and are spread across a gradient. For example, in human we don't just have tall or short people as two distinct alternatives, but a whole range of possible heights. Such traits are generally controlled by 3 or more genes and are thus called polygenic traits. Besides the involvement of multiple genes, polygenic inheritance also takes into account the influence of environment. Human skin color is another classic example for this. In a polygenic trait, the phenotype reflects the contribution of each allele, that is, the effect of each allele is additive. To understand this better, let us assume that three genes A, B, C control skin color in human with dominant forms A, B and C, responsible for dark skin color, and recessive form A, B, C for light skin color. The genotype with all dominant allele capital A, small b, capital B, capital C, capital C will have the darkest skin color, and that with all the recessive alleles small a, small b, small b, small c will have the lightest skin color. As expected the genotype with 3 dominant allele and 3 recessive alleles will have an intermediate skin color. In this manner, the number of each type of allele in the genotype would determine the darkness or lightness of the skin in an individual. Pleiotropy. We have so far seen the effect of a gene on a single phenotype or trait. There are, however, instances where a single gene can exhibit multiple phenotypic expression. Such a gene is called Chlorotrophic gene. The underlying mechanism of the pleiotropy in most cases is the effect of a gene on metabolic pathways which contribute towards different phenotypes. An example of this disease is phenylketonuria, which occurs in humans. The disease is caused by mutation in gene that code for the enzyme phenylalanine hydroxylase single gene mutation. This manifests itself through phenotypic expression characterized by mental retardation and a reduction in hair and skin pigmentation. Sex determination. The mechanism of sex determination has always been a puzzle before the geneticist. The initial clue about the genetic or chromosomal mechanism of sex determination can be traced back to some of the experiments carried out in insects. In fact, the cytological observation made in a number of insects led to the development of the concept of genetic or chromosomal basis of sex determination. Hanking could trace a specific nuclear structure all through spermatogenesis in a few insects, and it was also observed by him that 50% of the sperm received this structure after spermatogenesis, whereas the other 50% sperm did not receive it. Hanking gave a name to the structure as the X-body, but he could not explain its significance. Further investigation by other scientists led to the conclusion that the X-body of Hanking was in fact a chromosome, and that is why it was given the name X chromosome. It was also observed that in a large number of insects, the mechanism of sex determination is of exotype, that is, all X bear an additional X chromosome bodies beside the other chromosome autosome. On the other hand, some of the sperm bear the X chromosome, whereas some do not. Eggs fertilized by sperm having an X chromosome become female, and those fertilized by sperm that do not have an X chromosome become male. Do you think the number of chromosomes in the male and female are equal? Due to the involvement of the X chromosome in the determination of sex, it was designated to be the sex chromosome, and the rest chromosomes were named as autosomes. Grasshopper is an example of exotype of sex determination in which the male have only one X chromosome beside the autosome, whereas female have a pair of X chromosomes. These observations led to the investigation of a number of species to understand the mechanism of sex determination in a number of other insects and mammals, including man. XY type of sex determination is seen where both male and female have same number of chromosomes. Among the males, an X chromosome is present, but its counterpart is distinctly smaller and called the Y chromosome. Female, however, have a pair of X chromosomes. Both male and female bear same number of autosomes. Hence, the male have autosome plus XY, while the female have autism plus XX. In human being, in the Drosophila, in human being and in Drosophila, the male have one X and one Y chromosome, whereas female have a pair of X chromosomes beside autism. In the above description, you have studied about two types of sex determining mechanism, that is XO type and XY type. But in both cases, male produce two different types of gametes, A either with or without X chromosome, or B some gametes with X chromosome and some with Y chromosome. Such type of sex determination mechanism is designated to be the example of male heterogamy. In some other organisms, example bird, a different mechanism of sex determination is observed. Figure. In this case, the total number of chromosomes is same in both male and female, but two different types of gametes in terms of sex chromosome are produced by females. That is, female heterogameity. In order to have a distinction with the mechanism of sex determination described earlier, the two different sex chromosomes of a female bird has been designated to be the Z and W chromosome. In this organism, the female have one Z and one W chromosome, whereas male have a pair of Z chromosomes beside the autosome. Sex determination in human. It has already been mentioned that the sex determining mechanism in case of human is XY type. Out of 23 pairs of chromosomes present, 22 pairs are exactly same in both males and females. These are the autosomes. A pair of X chromosomes are present in the female, whereas the presence of X and Y chromosome are determinant of the male characteristics. During spermatogenesis among male, two types of gametes are produced. 50% of the total sperm produced carry the X chromosome and the 50% has the Y chromosome beside the autosomes. Female, however, produce only one type of ovum with an X chromosome. There is an equal probability of fertilization of the ovum with the sperm carrying either X or Y chromosome. In case of ovum fertilizes with a sperm carrying X chromosome, the zygote develops into a female XX, and the fertilization of ovum with Y chromosome carrying sperm result into a male offspring. Thus, it is evident that it is the genetic makeup of the sperm that determines the sex of the child. It is also evident that in each pregnancy there is always 50% probability of either a male or a female child. It is unfortunate that in our society women are blamed for giving birth to female children and have been ostracized and ill-treated because of this false notion. Sex determination in honeybees. The sex determination in honeybee is based on the number of sets of chromosomes an individual receives and offspring formed from the union of a sperm and an egg develop a female queen or worker, and an unfertilized egg develop as a male drone by means of parthenogenesis. This means that the male have half the number of chromosomes than that of a female. The female are diploid having 32 chromosomes and male are haploid, that is having 16 chromosomes. This is called as haplodiploid sex determination system and has special characteristic features such as the male produce sperm by mitosis. They do not have father and thus cannot have sons, but have a grandfather and can have grandsons. However, how is the sex determination mechanism defined in the birds? Is the sperm or egg responsible for sex of the chicks? Mutation. Mutation is a phenomenon which results in alteration of DNA sequence and consequently result in change in the genotype and the phenotype of an organism. In addition to recombination, mutation is another phenomenon that leads to variation in DNA. As you will learn in chapter 6, one DNA helix runs continuously from one end to other in each chromatid in a highly supercoiled form. Therefore, loss, deletion or gain, insertion or duplication of a segment of DNA. The result in alteration in chromosomes. Since genes are known to be located on chromosomes, alteration in chromosomes result in abnormalities or aberrations. Chromosomal aberrations are commonly observed in cancer cells. In addition to the above, mutation also arises due to change in single base pair of DNA. This is known as point mutation. A classic example of such a mutation is sickle cell anemia. Deletion and insertion on of base pair of DNA causes frame shift mutation. The mechanism of mutation is beyond the scope of this discussion at this level. However, there are many chemical and physical factors that induce mutation. These are referred to as mutagens. UV radiation can cause mutation in organisms. It is a mutagen. Genetic disorder. Pedigree analysis. The idea that disorders are inherited has been prevailing in human society since long. This was based on the heritability of certain characteristics, features in families. After the discovery of Mendel's work, the practice of analyzing inheritance pattern of the traits in human beings began. Since it is evident that control process that can be performed in pea plant or some other organism are not possible in case of human beings. Study of the female history about inheritance of a particular traits provide an alternative. Such an analysis of traits in a several of generation of a family is called pedigree analysis. In the pedigree analysis, the inheritance of a particular trait is represented in the family tree over generation. In human genetic, pedigree study provides a strong tool which is utilized to trace the inheritance of a specific traits, abnormality or disease. Some of the important standard symbols used in pedigree analysis have been shown in figure. As you have studied in this chapter, each and every feature in any organism is controlled by one or other gene located on DNA present in the chromosome. DNA is the carrier of genetic information. It is hence transmitted from one generation to the other without any change or alteration. However, change or alteration do take place occasionally. Such an alteration or change in the genetic materials is referred to as mutation. A number of disorders in human beings have been found to be associated with the inheritance of change or altered gene or chromosome. Mendelian disorder. Broadly, genetic disorder may be grouped into two categories: Mendelian disorder and chromosomal disorder. Mendelian disorder are mainly determined by the alteration or mutation in the single gene. These disorders are transmitted to the offspring on the same lines as we have studied in the principle of inheritance. The pattern of inheritance of such Mendelian disorder can be traced in a family by the pedigree analysis. Most common and prevalent Mendelian disorders are Hemophilia, cystic fibrosis, sickle cell anemia, color blindness, phenylketonuria, thalassaemia, etc. It is important to mention here that such Mendelian disorder may be dominant or recessive. By pedigree analysis, one can easily understand whether the trait in question is dominant or recessive. Similarly, the trait may also be linked to the sex chromosome as in case of hemophilia. It is evident that this X-linked recessive trait shows transmission from a carrier female to male progeny. A representative pedigree is shown in figure for dominant and recessive traits. Discuss with your teacher and design pedigree for character linked to both autosome and sex chromosomes. Color blindness. It is a sex-linked recessive disorder due to defect in either red or green cone of eye, resulting in failure to discriminate between red and green color. This defect is due to mutation in certain gene present in X chromosome. It occurs in about 8% of male and only about 0.4% of female. This is because the genes that lead to red-green color blindness are on X chromosome. Male have only one X chromosome and female have two. The son of a woman who carries the gene has a 50% chance of being colorblind. The mother is not herself colorblind because the gene is recessive. That means that its effect is suppressed by her matching dominant normal gene. A daughter will not normally be colorblind unless her mother is a carrier and her father is a colorblind. Hemophilia. This X-linked recessive disease, which shows its transmission from unaffected carrier female to some of the male progeny, has been widely studied. In this disease, a single protein that is a part of the cascade of protein involved in the clotting of blood is affected. Due to this, in an affected individual, a simple cut will result in non-stop bleeding. The heterozygous female carrier for hemophilia may transmit the disease to son. The possibility of a female becoming a hemophilic is extremely rare because mother of such female has to be at least carrier and the father should be hemophilic, unviable in the later stage of life. The family pedigree of Queen Victoria shows a number of hemophilic descendants as she was a carrier of the disease. Sickle cell anemia. This is an autosome-linked recessive trait that can be transmitted from parent to the offspring when both the partners are carrier for the gene or heterozygous. The disease is controlled by a single pair of allele H small b capital A and H capital H small b capital S. Out of the three possible genotypes, only homozygous individual for capital H b small b capital S show the disease phenotype. Heterozygous capital H small b capital A capital H small b capital S individual appear apparently unaffected, but they are carrier of the disease as there is 50% probability of transmission of the mutant gene to the progeny, thus exhibiting sickle cell trait. The defect is caused by the substitution of glutamic acid. HBA peptide, val his live tro glue glow. HBS peptide, val his live tro val glue. Glue by valine val at the 6th position of the beta globin chain of the hemoglobin molecule. The substitution of amino acid in the globin protein result due to single base substitution at the sixth codon of the beta globin gene from GAG to GUG. The mutant hemoglobin molecule undergoes polymerization under low oxygen tension, causing the change in the shape of RBC from biconcave disc to elongated sickle cell structure. Phenylketonuria. This inborn error of metabolism is also inherited as the autosomal recessive trait. The affected individual lack of an enzyme that converts the amino acid phenylalanine into tyrosine. As a result of this, phenylalanine is accumulated and converted into phenylpyruvic acid and other derivatives. Accumulation of these in brain result in mental retardation. These are also excreted through urine because of its poor absorption in kidney. Thalassaemia. This is an also autosome-linked recessive blood disease transmitted from parent to offspring when both the partner are unaffected carrier for gene or heterozygous. The defect could be due to either mutation or deletion, which ultimately result in reduced rate of synthesis of one of the globin chains, alpha and beta chain, that make up hemoglobin. This causes the formation of abnormal hemoglobin molecule resulting into anemia, which is characteristic of the disease. Thalassaemia can be classified according to which chain the hemoglobin molecule is affected. In alpha thalassaemia, production of alpha globin chain is affected, while in beta thalassaemia, production of beta globin chain is affected. Alpha thalassaemia is controlled by two closely linked genes, HbA1 and HbA2, on the chromosome 16 of each parent, and it is observed due to mutation or deletion of one or more of the four genes. The more genes affected, the less alpha globin molecule produced. While beta thalassaemia is controlled by a single gene, HBB, on chromosome 11 of each parent and occur due to mutation of one or both genes. Thalassaemia differs from sickle cell anemia in that the former is a quantitative problem of synthesis to few globin molecules, while the latter is a qualitative problem of the synthesis in incorrectly functioning globin. Chromosomal disorder. Chromosomal disorder, on the other hand, are caused due to absence or excess or abnormal arrangement of one or more chromosomes. Failure of segregation of chromatids during cell division cycle result in the gain or loss of a chromosome called aneuploidy. For example, Down syndrome result in the gain of extra copy of chromosome 21. Similarly, Turner syndrome result due to loss of X chromosome in the human females. Failure of cytokinesis after telophase stage of cell division result in an increase in a whole set of chromosomes in an organism, and this phenomenon is known as polyploidy. This condition is often seen in plants. The total number of chromosomes in a normal human cell is 23 pairs. Out of the 22 pairs are autosomes and one pair chromosome are sex chromosomes. Sometimes, though rarely, either an additional copy of chromosome may be included in individual or an individual may lack one of any one pair of chromosomes. These situations are known as trisomy or monosomy of the chromosome, respectively. Such a situation leads to very serious consequences in the individual. Down syndrome, Turner syndrome, Klinefelter syndrome are common examples of chromosomal disorder. Down syndrome. The cause of this genetic disorder is the presence of an additional copy of chromosome number 21, trisomy of 21. This disorder was first described by Langdon Down. The affected individual is short stature with small round head, furrowed tongue and partially opened mouth. Palm is broad with characteristic palm physical, psychomotor and mental development is retarded. Klinefelter syndrome. The genetic disorder is also caused due to the presence of an additional copy of X chromosome resulting into karyotype of 47 XXY. Such an individual has overall masculine development, however a feminine development of breast that is gynecomastia is also expressed. Such individuals are sterile. Turner syndrome. Such a disorder is caused due to the absence of one of the X chromosome, that is 45 with X0. Such females are sterile as ovaries are rudimentary besides other features including lack of